I'd like to welcome you all again for coming to what's now the final lecture in the series. So thank those who've come for every single time, which is great. And I think at this time, because we're the last one of the series, I'd like to also thank all the people who have kind of worked behind the scenes, kind of finding cables, setting up projectors, helping with visas to get Anab into Austria. So Nico and Peter and Matthias and Stefan, thank you very much. And the students for setting up all the refreshments afterwards. So that's been great. So thank you. So I think it's been quite an interesting journey through all of the different lectures we've heard. You know, lots of different issues and ideas have been raised. And I think from the very start, at the very beginning, with David and Sonia, we could see very quickly that it was very easy for the discussions to become very polarized between sort of positive things and negative things, you know, good and bad. It was very easy to do that, but to bring discussion into a central space that were a bit more difficult is difficult. So, but Kobe and Gunther, you know, from them we really looked into the world of DIY bio and what's sort of happening there, and how there's this sort of great interest in participating, and how the non-expert can somehow get involved and be part of the process of the changes that are going on with biological systems. So also with Kobe, he sort of introduced the idea that, that even, getting our hands into playing with the technology ourselves, there's still sort of online services that without any sort of abilities, you can kind of log on and access databases and information about these processes that we could never do before. So, this whole world is sort of really shifting and changing. And with Bruce and Marcus's lecture, we really kind of looked at how science stories, how they sort of start to seep into our everyday imagination, and in many ways they used, it's kind of, again, it's easy to use them negatively as fear tactics to sort of raise, sort of, create sort of negativity and the development of things. But it's very hard, I think, the science to balance between the hype and the promises that the technology is going to deliver us with a genuine kind of desire and enthusiasm to really engage and, again, participate in this. And I think Revital and Jens Hauser kind of picked up on all the work that was shown there, really picked up on that. So tonight in our final session, we have Anab Jain and Daisy Ginsberg. The title of this session is really kind of Futures Design Meets Science. And what I find particularly interesting about both Anab and Daisy is this: they find themselves sitting between many different disciplines and many camps, and in many ways they become a node in between, a kind of conduit between different practices. And I think they take design processes into places perhaps where design would never normally sort of sit comfortably, and so that's quite a challenging kind of space to be. So they've got this amazing entrepreneurial spirit, both of them, to do what they do and get out there and sort of ask questions in the ways that they do, I think is amazing. They're sort of new kinds of design explorers. So we're going to do the same kind of situation, 30 minutes, 35 minutes each presentation, and we can open up for discussion, questions immediately straight away and see if we can have a discussion. So I'd like to introduce initially, first of all, Anab, who is director of a company in London called Superflux. I first met Anab, I think, in Microsoft Research Labs when she was doing work with robotics. Was it the robotics group? She in their office, they do lots of self-initiated work, as well as sort of working with clients like Microsoft and Sony, BBC, Princess Foundation. So really mixed practice, and she's a TED fellow, and in both Daisy and Anab have won various kind of awards. Anab in Apple Computers, UNESCO and ICSID, she won awards there. Work has been presented in MoMA and China, and is always a great conference speaker on conferences like Lift, Picnic and SIGGRAPH. So actually there's a great conference you did in Berlin, which I'll send out to the students later, which is a slightly different one, which again, very provocative. So I'd like to thank very much indeed Anab.
Thank you. Thanks, Fiona, for inviting me. I actually really love Vienna because I did 10 years ago, shows how old I am, I studied here at the Academy of Fine Arts, and I think it's the most beautiful city in Europe. So I'm really happy to be back here, even if it's just for one evening. So thank you. So yeah, I'm just going to talk a little bit about our studio and just kind of tie in some of the projects with you, which we do with the themes that you've been exploring around biotechnology, synthetic biology, and design. This was a sketch we made for the studio a few years ago, and we kind of maybe found a sort of way in which to present ourselves. So we call ourselves, we do both consulting, and we are also a creative lab. We work for small and big organizations to help them think creatively through various sort of weak signals around the futures and translate them into tangible products and services. But we also have a lab through which we do self-initiated work, and through that self-initiated work, we are able to push the boundaries around things we are interested in without having the pressure of a client. And it's great because both the sort of sides feed into each other, and quite often you have the thing in the middle which we call the sweet spot, where a product becomes part of a speculative research, or some of the speculative research moves into becoming more of a product. And so that sweet spot moves between the sort of now and the future. And essentially we say that we design for the imminently probable, things that are likely to happen, things that might be plausible. How do you realize them? What are the sort of experiences you design and the stories you tell to help people suspend disbelief and enter those worlds? So some of the projects that we work on might involve doing digital products around the Internet of Things, augmented reality, designing prosthetic vision, or even playing with experiences around civilian drones. So it's a very broad practice, but at the heart of it is the idea of how do we humanize technology, and how do we sort of create a more collaborative space for people to work around technology. The studio was founded by John Arden, who's also a designer at the graduate from the RCA, and myself. And we like to work as a sort of team of collaborators. So Rafael's actually here. He works with us off and on. We prefer to work with people who come in and out of the studio pretty much like a film production team. So you're not, nobody works there all the time. Everybody has ownership of the project. Everyone is free to go and work on their own projects when they feel like. And there's a sense of a trust between the things you do. And it's really helped us grow by having this sort of a slightly loose structure. We work mostly in the UK, but I'm from India originally and have family there, so I like to keep going back. And so we also do projects in India. For instance, one project I did a few years ago around augmenting pets with RFID chips, and even creating this idea of Internet of Pets. Similarly, the idea in India would be to use the ubiquitous cow and create a MUFI, or a mesh network powered by cows. Obviously not for real, but the idea that you sort of start looking at the things within the local context. But in a way, this idea of augmenting pets or playing with nature is a theme that runs through some of our work, and that's what I'm going to focus on today. So I think through all the stuff that you probably have heard in the lecture series, this question might have come up a lot about what is real nature, for instance, this image is of a land being farmed. Is farming natural? Looking at this sort of an industrial scale farm, one begins to wonder what it means to have something called natural. And Bruce Sterling wrote a really nice article in an online journal called Nature, where he writes about how nature is being branded. So he says this vaguely politicized attitude about nature never came from nature. It was culturally generated. Nature didn't get her all-natural identity branding until the Industrial Revolution broke out. Then poets and philosophers were allowed to live in dense, well-supplied cities where they could recast nature from some intellectual distance. Before that huge effusion of organized artifice, people lived much closer to the soil. And this sort of idea of how increasingly our own bodies and those of animals are getting intertwined, the materials that we play with, our blood and DNA, is increasingly raising the question of what is design, can nature be design, and what are the sort of ethical considerations around that. And it becomes even more prominent because everything that you see in the news, a lot of things are headlines such as this: genetically engineered mosquitoes to invade Florida, where this company, a British firm called Oxitec, is trying to modify mosquitoes so that they are able to save people from dengue fever. Or infecting mosquitoes with bacteria could block malaria. So they found a bacteria that reside in insect cells and have a very complicated relationship with their hosts. They can render mosquitoes resistant to certain pathogens, and in that sense, they will reduce the mosquito's lifespan. So you have engineered bacteria to destroy mosquitoes. And of course, this thing became a big headline last year. Mad scientists create artificial jellyfish from rat hearts and a sheet of silicon. And this is actually that jellyfish thing sort of starting to come to life, apparently. Well, obviously it was a piece of silicon, but they're very proud of this work of having this sort of artificial jellyfish created in the lab. Recently you heard about this thing called the rodent mind meld. Scientists wire 2 rat brains together to share sensory information in a major step towards what the researchers call the world's first organic computer. Wireless implantable interface lets monkeys control computers with their brains. It only gets better. Recently, there was this big Kickstarter project that went way over its target. Glowing plants, natural lighting with no electricity. Absolutely, the guy managed to get great sort of quotes from very, very famous people. He will send you the seeds of this plant that is synthetically engineered, apparently, and you can plant it and it'll start glowing, apparently. I'm highly suspicious about this whole thing, but the fact is that I don't even know what the laws are, because actually there are very stringent laws around what you can and cannot grow. So it's quite interesting that something like this can get so much funding through a platform like Kickstarter. And this is not just at a small scale. So on the completely other end, at an industrial level, there's a lot of very serious work going on in this space. So I'd like to show an excerpt from a documentary called DNA Dreams, where this filmmaker goes to BGI, a big genomics group in Shenzhen, China, where they do a lot of cloning around pigs and animal cloning.
Trying to make these pigs more happy and more released, we just put the shoe coat on its legs, and then just let it lie like this.
So are they making a transfer right now? Or?
Yeah, they're making the transfer. They take this uterus out and then make a small hole on the uterus cone, and then inject all these blastocysts inside.
Yes, genetically designed blastocysts. So.
Yes. Hopefully this cell will get pregnant.
And you say they are doing 2 a day? Or how?
Normally they did they transfer 3 recipients every day.
3 recipients?
Yes.
In Denmark we make perhaps 12 transfer in a whole year. And, and you do that in 4 days?
Yeah.
You're better than nature now. The success rate is really impressive.
So this is our new stables.
Okay. So when will this be ready for use?
In 1 week.
In 1 week, okay, yeah.
That's what we call Shenzhen speed.
Yeah.
We do everything very fast. Here we only do the pig cloning, and we have planning another cattle cloning lab in Shandong province. And now there are several people that are working on the sheep cloning in Xinjiang province.
Yeah, Shenzhen speed genetic modification is brilliant. It's quite interesting to see BGI scientists, now researchers, being brought to universities across Europe and America to give their expertise on how things can start getting faster here. So there's a lot of activity in this field. In fact, these could be the world's smartest babies. Again, a project by BGI, who have collected DNA samples from 2,000 of the world's smartest people and are sequencing their entire genomes in an attempt to identify the alleles which will determine human intelligence. Apparently, they're not far from finding them, and when they do, embryo screening will allow parents to pick their brightest zygote and potentially bump up every generation's intelligence by up to 5 to 15 IQ points. On the other hand, closer to home, you have the company 23andMe, which you probably might have heard about. Over the last 6 years, they've collected 180,000 people have sent their saliva samples using their cute spit kit in exchange for valuable health and ancestry information. But recently, founder Anne Wojcicki, who happens to be the wife of Google founder Sergey Brin, decided to monetize their giant database of genetic information. So they opened their treasure trove of genetics data to third-party developers to build new web-based interactive tools. Although current laws prohibit people, such as employers and insurance companies, from discrimination based on genetic information, life insurance providers, schools, athletic organizations can all potentially get their hands on your genetic data. And I can't even begin to imagine the impact this will have on dating apps and sites. So it's a bit like Atwood's world of Oryx and Crake, where these new stories of genetically modified animals and chimerical birds and creatures might sound a bit like a science fiction novel, but that's just because these are edge cases that challenge our ability to start sorting things out in neat categories that you might have seen. So you can't say this is sort of easy. Is it machine? Is it artificial? Is it natural? It's quite difficult to sort of do that anymore. And in a way, it's because of the materials and the access to the tools we are having that we're constructing not only ourselves, but what might be natural around us. Just like we might construct chipsets or political systems, we're creating a whole new genre of hybrid objects. And this sort of modification of biological organisms is not new. I mean, we all know, and you've probably seen examples, we've intervened with nature for a long time. I'm sure most of us can't even remember what wild chickens looked like. And these bananas, if you say, no, I'm not going to eat genetically modified food, surely everyone eats a banana. And bananas, as we know them, are a mutant strain. So probably every banana every person is eating is modified. And then, of course, these are the atomic seeds, which were hugely popular post-World War II, where people bombarded plants with radiation in hopes of producing mutant varieties of larger peanuts, disease-resistant wheat, more sugary sugar maples, and African violets with 3 heads. We also tried to make our own version of atomic seeds as an experiment to see if people would want to grow their own sort of atomic corn plant. And so it was an experiment that was quite interesting because people would not obviously go and start sowing the seed, but it became a conversation piece. And so I think the question is, why should we talk about it now? If it's been going on for years, it's been going on for centuries, this sort of modification with nature, why should we talk about it now? I think one thing is that technology and science today make it possible to not just manufacture the capability, but things are starting to be designed to grow inside our bodies, and that's that of plants and animals. It's possible to apparently edit and design living organisms is a whole different thing. It's a kind of thing that makes more and more people uncomfortable and confused. For instance, a fantastical animal, the spider-goat demon, as imagined by Albert Seba in his 18th century Cabinet of natural curiosities becomes real, we have no choice but to take notice. So again, I'm sure you've heard of Freckles, the first generation of transgenic goats. Before she was born, she had spider genes added to her genome, which caused her to produce spider silk protein in her milk. The protein can now be extracted and spun into silk, which is desirable for its incredible strength. So you have a whole industry of people breeding goats with spider silk protein in them. And I'm sure David must have definitely talked about iGEM, so the intensity with which these hybrids are being made and the easy access to changing technologies that we have to make them is also making a huge difference. iGEM has, I think, 1,500 undergraduate students participating every year. And I think last year they also introduced a strand for school students and entrepreneurs. So you need not be just a graduate, high school, a college graduate student to participate in iGEM anymore. And projects such as the E. coli bacteria as photoreactive film, or this thing which was made by, which might look like an art made by a child, but it's actually a representation of genetically modified bacteria that are created by isolating genes from organisms that have plastic degradation properties in them and inserting them into marine bacteria. This is the UCL iGEM team in London, and they worked with the London biohack space to come up with this idea. If they were to succeed, these new plastic-eating bacteria could be a natural solution for the millions of plastic bits floating in our oceans. And this team went ahead and started creating this idea. So they claimed the plastic republic in the middle of the North Pacific Ocean. And after iGEM was over, they continued working with the Biohack Lab in London to design what they are saying is the world's first public biobrick, where the DNA code was created and submitted to a parts registry outside of an academic institution. The biobrick they created could degrade mercury, which is a common water pollutant in India. Apart from iGEM, you have 300 plus DIY biolabs and enthusiasts around the world, showing how technologies that were the remit of scientists are now increasingly cheap and easy to access. So, obviously, I think a lot of us are grappling with this question: what will be the prototypes of this new 21st century nature? When what might be perceived as nature merges with science, technology, and politics, what sorts of new categories of things will be needed? And when the techniques to make them easier to access, who will be designing, manufacturing, and distributing these kinds of hybrid products? Like the cyborg was for the 20th century, what will be the prototypes of this new nature be? I think these are some of the questions we are interested in asking at Superflux through our own practice in different ways. And what I'm going to do, I'll just show you examples of dip into some projects that we've been working on to illustrate these ideas. So, contrary to going into a project and producing some work that would become led just by ourselves, I put a public call out for a project called Power of 8 a couple of years ago, inviting people to imagine alternate futures on Vimeo, sent it out, got lots of people who were interested. So it was a 3-month intense sort of studio with unknown people, not designers, like a nurse, a teacher, a permaculturalist, a technologist, a writer, and so on, to understand what does it mean, what are our sort of collective visions of the future outside of getting freedom from mortgage or planning exotic holidays. Over several workshops, we tried to build what we thought was an optimistic vision of the future, but what emerged was perhaps a little bit more problematic. A haunted alternate ecosystem of strange machines and modified nature. And that was partly because the main concerns that the people came out with were around climate change. Oh, I want a greener nature. I want to be near nature. I want greener streets. I want to sort of reverse this idea of climate change. And actually, it's far more complex than that. And so we decided to run with this idea of this alternate ecosystem and started creating the world around it. So we worked on 3 sort of main ideas. One was new pollinators, second was new weather, and third was the new economies that would emerge. So just quickly to say that none of these things were meant to become real working prototypes, but were meant to be stories to illustrate to a public, again, a suburb in London called Brentford. So we were working within the gallery and people would come in there all the time and discuss. So it wasn't meant to be, it was quite an open process again. So yeah, in this case, the idea was that you have these sort of autonomous flocking clouds for the residents of Acres Green, so that wherever there was less rain, you could sort of invite these clouds to come and form, make a formation and have rain. Obviously, it's a very sort of fantastical idea, but to be able to visualize this was very important for people who are non-designers, and to be able to question these ideas is equally important. So part of our process is also to be able to create these long-term discussions. One of the scenarios around the swarming clouds was that they had ultrasonic sensors, so they would attract bats, and they become a spectacle to watch out in the evenings, and so on. I think one of the women, I think she was a teacher, and she was really interested in the idea of the colony collapse disorder. The bees are disappearing, bees are a big problem. How can we sort of reverse this process? And usually you're always bombarded with the idea, save the bees, save the bees. And so we proposed to her that what if we could design a new kind of bee, a synthetic bee, or a beamer bee that would allow, that would be okay. So if there are no bees, we'll use these bees instead. And it was difficult for her to get her head around it. And one of the bio-nanotechnologists from Imperial College made the sketch for us to say how we could potentially have this beamer bee. And we kind of ran with it and imagined idyllic scenarios of people using these sorts of bees to grow food in their gardens, or following people on their way home on warm summer evenings, and so on. Even children who could keep glowing bees as pets. So she and a lot of other people sent us a lot of angry messages that, this is not right. In fact, she was like, I don't want to work on this project anymore. I'm like a supporter of nature, and you're just kind of going in this. But I think that was very important, that it's equally important to get people getting angry as it is to sort of people getting feeling positive about your projects was quite important for us. And then I suppose you also know about the fact that there are defense personnel around the world. So drones are big, and you know that they're all toying with the idea of using miniature drones disguised as bugs or insects for spying purposes. Apparently, DARPA's ultimate plan is to eventually hack into the insect's own natural senses, allowing the remote control operator to look out of the insect's own eyes instead of attaching a video camera for it to carry. So what's stopping them from making the world's first 100% natural drone, a synthetic bee, as a future surveillance device? And those are the sort of questions that start emerging from the project over a period of time. And then again, we went back, and just recently in The Guardian was the idea about this, why make tiny flying drones when you can fly real insects by remote control? So Emily Enthis has written a book called Frankenstein's Cat, which I really recommend everyone reading, in response to DARPA's open call. And this is when it starts becoming interesting, because lots of people find this quite terrifying, the idea that you are kind of really blending this sort of machine nature hybrid and starting to use it for purposes that were not originally, that they were not supposed to be originally designed for. In fact, a lot of scientists say that cockroaches are the perfect cyborgs. They are designed to be hacked, and nature has designed them to be hacked. You can read articles where they say that sort of thing. So I think this is where speculative design can be very useful as a genre, because it can help to create the alternate possibilities around such sorts of new technologies and juxtapose sort of alternate worlds with our lived realities today. Moving on to us as human beings, obviously we don't remain outside of this conversation where nature is being hacked and manipulated. And the idea of cyborgs is obviously on for a while, but would you call something like the Google Glass, someone wearing a Google Glass, a cyborg? That's a big question that a lot of people ask. And yes, there is a Tumblr, white men wearing Google Glass.tumblr.com. A lot of hype around this sort of ambient technology. Except that it's not it's quite interesting the way they sort of present the technology as a visual device that has been modified to look completely slick. But there's not that much conversation about what happens, what are you recording, and what if you get recorded, what happens to your own sense of privacy as a spectator. Anyway, the reason for showing this glass was that we are also working along similar lines to design new kind of augmented vision. The only difference is that we're using, working with scientists who are using gene therapy. So a ongoing project in the studio called Song of the Machine is about the design of prosthetic vision. The idea is to restore some form of vision to people who are suffering from retinitis pigmentosa. And it's possible to do the science of optogenetics, which is a combination of gene therapy and optoelectronic prosthetics. And what I will do is I'll just show you a video.
Patients with retinitis pigmentosa go blind because the light-sensitive cells in their eyes are dysfunctional. The remaining circuitry is, however, intact. In 2003, the discovery of a special light-sensitive protein in algae led to the new field of optogenetics. We can use these special light-sensitive proteins to bring back some form of photosensitivity in the remaining cell layers of the retina. My team is developing a special optoelectronic headset to interpret the world and communicate with these newly light-sensitized cells. It is our hope that with a mixture of optogenetic gene therapy and our headset that we can return lost vision to these patients.
So he's a scientist, and he's got this he's using optogenetics. They found this protein in an algae in a pond that is light sensitive, and they insert it as a virus behind the eye. And that kind of makes the dead retinal cells in the eye light sensitive. And then you wear a headset, and then you can start seeing some form of vision is restored. So it's almost like plugging the SCART lead to your head and starting to sort of really play with your sort of sense of reality starts shifting quite significantly. Interestingly enough, these scientists were trying to create these sort of very crude image augmentation concepts and testing out with their a group of people that they were working with, their sort of user group, cartoonized views and chromatic edge simplified view and a Tron vision and whatnot. But what people came back with was quite interesting. A lot of people said, I'd rather be blind than have my world look like this. And that was quite interesting because they were so convinced that if someone is blind, they would be happy to see whatever that vision is. While that's not the case, it's the same with people who are hard of hearing. They do not want to restore. So this is a very interesting thing that a very data-centric perspective is not going to solve. So that's where, as designers, we are often asking the question, what will be our experience with technology and what will it feel like? So kind of human centricity over efficiency. And so it was great to sort of start working with scientists to start zeroing in on what's preferable as opposed to what's possible. So that's the space that we found quite interesting and started working with the scientists. So just through the process of working with them, we realized that actually the technology could allow people to start seeing in a much wider electromagnetic spectrum that is not normally visible to the normally sighted people. So far infrared, near infrared, all the way to the UV, as well as augmented reality could be accessible to people who've gotten blind because of retinitis pigmentosa. And that was fascinating. So what if you could shift your vision between the different spectrums depending on where you were? What if you could start seeing nature through the eyes of bees and butterflies through ultraviolet? That was quite fascinating. And so very quickly we kind of created some very quick props, intelligent headset, a handheld gestural device with tactile fiduciary markers alongside the person to allowing the person to send messages to the headset. And then I'm just showing you some excerpts from the film that we made, essentially as a conversation piece for the scientists to start understanding a range of possibilities. Yeah, so this is just one excerpt. We kind of went through the different modes. In this case, it's near infrared and makes nature and green, like leaves and trees appear very snowy, completely different sort of sense of the world. And another quick clip. Yeah, so again, more explorations. And this was obviously a piece that kind of the scientists were quite intrigued by, but it's going to take a while for this sort of a vision to be possible. Initially, it's going to be quite low resolution. And what was interesting is that we are now able to move on to the next step, which was to continue the relationship of working with the scientist in a much more sort of, it doesn't remain just a collaboration where you meet the scientists a few times and then go away. You actually work together with them. You keep understanding. So you're developing. What is fascinating for us is how can we help develop the technology before it becomes a product. The shaping of the technology before it becomes a product is very fascinating. And so now the idea that what is the actual vision, what are the apps going to be, we have done a lot of research with people who are in different stages of retinitis pigmentosa, and what are the sort of functional things that they need, facial recognition, being able to feel comfortable in social situations, being able to navigate the world, being able to kind of see beyond your kind of, because there's a very limited view. So how do you see peripheral vision? How do you avoid obstacles? These are very pragmatic things, but how do you make them in a way that they are a bit more poetic, a bit less about this constant voices telling you turn left and turn right? And that's an equally interesting challenge and might take a long time for us to kind of get there. The second thing about this is the sort of ethics around this project. Because of working with the scientists on a longer term period, we get a real insight into how gene therapy occurs, what are the sort of ethical considerations for any such thing, what are the rules and regulations in Europe around creating such experiments. So for instance, last year, for every year, but I think definitely last year, they used 30 mice, 15 rats and 5 rabbits per year for the experimentation. This year they've got special monkeys that they've got from Africa, I think, to Paris, because they were not allowed to bring them to UK. And they have to go through several hurdles before they can actually start working on this project, which I think is also important for us as designers to be involved in. And then once we start looking into it, there's a whole industry that has actually sprung up to sell diseased lab animals to scientists. Scientists need to have different kind of mice so that they can see over periods of time. So there was a fair in 2011 in Florida where all the biotech manufacturers from China especially came around, and they had booths in a ringing ballroom advertising animals that had been engineered to suffer from things like cancer and cystic fibrosis. A brochure, for instance, said, you design the experiment, we'll design the mice. I mean, I was just talking to Daisy, who's got a much more extreme example. She'll probably talk about her experience from Symbiotica as well. There's a company called Exemplar Genetics. If you start going into their website, it is pretty freaky, the sort of pigs that they are designing. To then sell to the research labs. And this is very important research at one level. They are trying to find cure for cancer. But there are a lot of very interesting sort of edge boundary sort of cases where you're not entirely sure, is this right, is this ethical, should they be doing this or not? And I think that's quite important. Also, you've heard in the news similarly things that have started coming out about code. For this is about myriad genetics, that they were, Supreme Court recently allowed them to patent human genes. They specifically found 2 genes, the BRCA1 and 2, which can mutate in ways that carry a high risk of breast cancer and ovarian cancer. And the thing is, what they were doing is they were trying to they wanted the patent for isolating the gene. Now, the argument of the company is that isolating the human gene is actually a man-made process, and it's a very important invention, and we need to patent it in order to continue innovation. But a lot of people feel that genetic material in the human body cannot be patented because it's part of our genetic makeup. If the consequences of this are interesting. So every patient who now wants to test for breast cancer or ovarian cancer has to go and use only Myriad Genetics test, which is very expensive, especially in America, costs $3,000. And so if you don't have the right kind of insurance provider, they won't even sort of spend that money for you to do the test. There's also more and more of this sort of thing coming out, that actually 20% of the human genome is under patent in the US. And in Australia, there are lots of patents as well. Very severe syndromes tests are now only being conducted in Europe, so a lot of children who suffer from these diseases, their tests are not being sent to Europe. And so the consequences of these things are quite interesting. And this is something we started exploring in a project recently called Dynamic Genetics versus Man. It's a project which imagines a world where synthetic biology and gene therapy have moved into the marketplace. In this world, the state's responsibilities have shifted from healthcare provision to the provider of health insurance. So by calculating the healthcare implications of specific gene combinations, insurance rates are adjusted on a person-by-person basis, ensuring that individual contributions accurately reflect the true potential costs associated with your genome. So if you have a risk of diabetes or cancer, it will be calculated in the algorithm, and you'll be paying your insurance premiums accordingly. What new laws and economic models might emerge under these conditions? How will intellectual property be applied and policed when designed genetic material makes its way into people's bodies and their lives? These issues and the wider implications are explored through the lens of a court case. So this is a company called Dynamic Genetics, and they have which is a large gene therapy conglomerate in this future world. And they have put a case against this guy, Arnold Mann, who they accuse of obtaining copyrighted DNA from a black market clinic. Who owns our genes? Who can patent them? Will we have patented children? And on the flip side, will we have pirated children? How would healthcare models adapt to these new changes? And ultimately, how will we value human life? I think these are all equally important questions that we have to ask while working in this field. And to do that, we designed a workshop tool called the SynBio Tarot Card, which is to help designers and scientists explore the wider social, economic, and political implications of synthetic biology. So it's a starting point for conversations around the implications. You can pick up each card, and there are things about various sorts of events going on. So for instance, you might be asked to pick up a card about that you are a celebrity, you are in Dubai, it's 2025, the Open Gene Directive has been issued, there is a breakup of nation states, and your mission is to help humanity make that happen. What are the things you have to consider? Especially interesting for the scientists who use this in the workshop, because their focus is on isolating this particular gene from this module, and very, 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 very specific narrow focus. But when they were pulled out into this wide world, it was quite an interesting thing to do. I'm not going to go through the scenario, but David, who spoke in the first instance, has actually put it online, so you can go and have a play with it yourself. So I suppose, in a way, it's more about trying to think of a more liquid approach to design. Genes express, and we cannot tell what the mutated products will look like. It's about kind of thinking how we can embrace mutations, how can we use this opportunity to slow down and be more deliberate and conscious about what's happening around us. How can we situate ourselves within the legal, political, and social implications when blood and DNA become our sort of new materials. Yeah, I think I'll end on that. Thank you.
So actually, I think
Sure.
Daisy sits really in the centre of the whole SynBio kind of. She's found herself in the middle, and I remember at the very early stage where Bjorn Frank, me and Daisy went to a conference, was it in 2008, 2009, full of synthetic biologists. I think we were the only non-scientists in the whole conference, and they'd all thought we'd got lost. But it was our introduction to this whole world in synthetic biology, and in many ways from that moment, Daisy's continued on really trying to understand what's going on in this space from a perspective as a designer. She has been involved very, very closely with some of the key scientists working in this area through a fellowship that she did with Stanford University and the University of Edinburgh called Synthetic Aesthetics. It's now going to be, is part now of a book that's being published by MIT Press, which comes out in 2014. So do look out for it. I think it's going to be one of those books that really sort of, marks a moment of all the things that are kind of happening around this at this time. But apart from that, as a designer, she's been recognised in the world of design. So she was nominated for Design of the Year Award and Index Awards, and received the World Technology Award for Design, and in 2012 won the London Design Medal for Emerging Talent. So I think that's really interesting that her work is from the design world is getting recognition. So.
Yes, I know about synthetic biology, but not about digital technology at all. So thank you so much for the invitation to speak here this evening. It's really, I haven't been in Vienna since Marcus arranged the Biofiction Film Festival, so I know only the Natural History Museum here. So today I'm going to talk about what I see as the role of design in biotechnology, and my research for the last 6 years has been looking really specifically at this question about what is design in a so-called biotech revolution, and how might design change. So let's start with something very simple that we know. Before the 1970s, you'd rarely see one of these. The supermarket became part of everyday life, and shoppers came to expect the disposable convenience of a plastic bag. The plastic bag in its very humble way is just a thing of progress. Plastic bags were better than paper bags. They were cheaper, stronger, they used less material to make them, and fewer trees are cut down to make plastic bags than paper bags. They're environmentally friendly. So much so, in 2001, we used 1 trillion of them worldwide. Plastic bags themselves are part of the problem, but it's our use of them that is the real problem. Although we know that plastic isn't disposable, we carry on using them every day. Another use of plastic we've come to accept is the water bottle. We know that plastic doesn't disappear after we're finished using it, yet in 2011, Americans drank 34.4 billion litres of bottled water, and that was 4% more than in 2010. It's a recession-busting industry if ever there was one. The plastic bottle is safer than glass, less carbon is used as it's transported. Plastic bottles are better. But are they, really? We solved the problem of convenient drinking water, but we made a massive new problem of waste. And yet, we all know this, and we all use them every day. The incandescent light bulb. We replaced it with something better. The energy-saving light bulb. They use less energy, they last for longer. But while we threw the old ones out, these ones we have to recycle. And we recycle them, or we're meant to, because they contain mercury, which leaches into the ecosystem. And yet, many people still throw them away because it's easier. Our behaviors are difficult to change. So is this really better? Well, it's complicated, and this is a very simplistic argument, but the plastic bag, the plastic bottle, and the so-called energy-saving light bulb are all really simple, everyday examples of how design and technology and engineering can take a problem and solve it, and give us more problems, all in the name of progress. We assume that design makes things better. We take for granted that's what we do as designers. We improve things, we make a plan and we do it. But what do we mean by better? Do we mean longer lasting, cheaper, more high-tech, more sustainable? Better for whom? Whose better shapes our common future? We keep on doing the same thing over and over again as designers, making plastic bags and bottles and light bulbs. And what do we often call that kind of behavior? Well, insanity. Einstein said a lot of clever things. This one's just attributed to him. I think it's actually from Narcotics Anonymous. But he did say this: we can't solve problems by using the same kind of thinking that we used when we created them. Yet we keep on doing the same things with design. As designers, I think we must and we can and we should do more than just solve the problems that are brought to us. We need to challenge those problems and use design to ask different, more relevant questions. This is what I learned in design interactions. The question now is, can design help us understand what we mean by better? So this evening in this talk, I'm going to ask, how might we use design to ask better questions in places where we don't even know what the questions are yet, and so that we don't just solve the wrong problems? Because engineering is about solving existing problems. Design, in theory, is about possibility, about life as it could be. Design projects into our future and makes new possibilities out of existing matter. But designers want to be problem solvers, like engineers. This is why I'm not a designer, I'm probably an artist. This has been kind of the problem all the way through for me. And I think we need to get back to this idea that design can help shape our future. Design can actually be about possibility. So it's to the future that we're going to look now, and how design can actually work upstream in the development of new technologies, where futures are being built and constructed, like all the things that Anab's talking about this evening. How can we use design to actually ask better questions in these technologies and actually use design to affect the direction a technology takes before we even get to problem solving? Can design help us alter the future? So I'm going to share some of my experiments in this area, and I'll tell you a little bit more about synthetic biology. I think everyone probably knows it well by now. And then I'll tell you about some of the projects that design different ways of thinking and some of the collaborations that I think hint at new ways of working for design. So I began in architecture, and then I got distracted by this. This is a plate of agar jelly. I'm sure you've seen a few renderings of this in different ways. And Anab showed one. So it's covered in E. coli bacteria that were engineered by undergraduates to turn dark when they're exposed to light, like photographic film. And I find this really interesting because, like all good biological design projects for iGEM, it has to be given a comedy name. So this is E. coliroid, and Hello World is a reference to the first computer program that you learn to program. But obviously this is in biology. This is the very beginnings of synthetic biology for many students. And the difference between genetic engineering and synthetic biology is one of ideology. So GM, this is a flounder, you take an antifreeze gene from this fish and put it into a tomato to make that tomato resistant to cold. But the key thing here is that it's a one-off solution. For the synthetic biologists, they see the genetic engineer doing this and like, oh, it's like making a bespoke suit. But synthetic biologists want to be able to take a gene from that fish and put it into any number of organisms and be able to be sure of what the results will be. They're trying to make biological engineering more like computer science, standardized, predictable, repeatable. That's not what biology is. Biology lives and dies and reproduces, and it's completely different to the binary rules of computer logic. And that's the challenge for the synthetic biologists. Luckily not for me. But as synthetic biology promises to turn engineered biology into this material that could solve all our problems, they're saying they're going to make the world better. Materials, fuels, medicines, food, biology, and life with it is simply being transformed into a 21st century material for design. So what I've been asking is synthetic biology just a design discipline of the future? Perhaps. But at the moment, as an emerging field, there's been very little precedent for design or role for design, and that's why I think it's such an interesting area to test ideas as the kind of concepts and the disciplines are being shaped themselves. Because at the moment, it's about problem solving, making big vats of stuff like engineered yeast to make jet fuel, or rubber to make bacteria to make isoprene for rubber tires, or non-biodegradable plastics. The same things that we already have. What could be a disruptive technology is going to disrupt nothing. And that's part of the appeal for the people funding it. We can make the same fuel and operate in the same systems. But I think we should ask not only how do we make yeast make jet fuel, which is a problem for the scientists to solve, but we need to ask different questions. How do we design ways to use less fuel or alternatives that use no fuel, rather than just going in the same linear direction that we assume that progress runs in? I think this is where we'll find innovation that's better. Instead of just making technology, we need to be asking why we're making it, what we want from it. And perhaps this is where we'll find novel ethical innovation in synthetic biology. So how do we apply this kind of thinking to the real world? This is something I've been experimenting with now for 5 or 6 years. And at the end of my master's at the RCA with Fiona, I was talking about system collapse with, I think it was the end of being a student, with Sasha Poflep, another design student there. We were talking about in a future where big systems collapse, like peak oil, this idea that we're going to run out of oil in sometime in the 21st century, could that kind of thing force a technology into action? Could we imagine that synthetic biology would be forced into action and we maybe see disruptive manufacturing systems that are made by biology? As we were having these conversations, we found out about the work of Jim Hasselhoff, who's a plant scientist at Cambridge. And he researches plant morphogenesis, the way that cells divide and differentiate, and potentially how they could be programmed and controlled. So I wrote to Jim. And this is he was probably the first scientist who actually replied to one of my emails after many false starts. I went up to visit him in his lab. He started showing me this. This was footage of a carnivorous pitcher plant that grows its own vessel to capture its food. At this point, I had thought that synthetic biology was just about bacteria and microbes and yeast growing in vats. But clearly, there was something else going on. So I asked Jim, where do you see synthetic biology with plants going? What should we be imagining? Jim said, well, maybe one day we'll grow products inside plants. It was deliciously ambiguous, because did he mean like chemical products, like sort of genetic modification as we know now, or was he really hinting at controlling form, like this pitcher plant video that was playing on his desktop? So I went back to London, got the train, told Sasha all about this, carried on with this conversation about disruptive technologies, and we wondered maybe synthetic biology with plants would be the way that this would be solved. We could program them to solve this problem, our consumption patterns that are completely unsustainable, where we ship stuff backwards and forwards and all around the world before and after we've consumed products. And that would, in this scenario, would mean living organisms as industrial robots, replacing the mechanical production lines that we have today. And this became growth assembly. So growth assembly is a product that we designed from this potential future. And we designed it to understand what we were talking about better, and it was really sort of investigation, not prediction. So growth assembly are 7 parts of a product which are harvested and assembled to make a herbicide sprayer. And we chose this as our everyday product from this future, because we thought everyone would need a herbicide sprayer. Everyone's growing their machines out of plants. You need to protect those horticultural machines from nature that can sort of naturally defend itself. So it grows in, this is the herbicide gourd. It's like a big melon, and you put your arm through it and you'd squeeze the juice out. The tubes are grown as long, translucent roots, and we were taking advantage of gravity here to make them sort of grow further. You could move the water down. And I was emailing Jim during this process, saying, so Jim, we're doing this really weird project now. Any tips? He's like, well, maybe, you should, he sort of gave this, I've got this lovely scientific email about the constraints of morphological systems, and sort of evenings in the pub trying to work on this with the illustrator. Quite good. These are nozzles. They're grown in a fruit related to the tomato and picked off the vines. And the idea is that grown, harvested, and assembled, it would change our expectations of the things that we manufacture. Softness and diversity would replace hardness and manufacturing standards that we expect today. Shops would evolve into factory farms. Licensed products would be grown and sold in the same place. We'd no longer need to ship things around the world. Instead, we'd just ship seeds, because all the manufacturing instructions would be encoded into their DNA. So we're very pleased with ourselves. We solved the world's problems. The technology the idea is seductive, and potentially, who knows if it could work. But there's something I find that's really troubling in this project, is that biology is completely industrialized. And we've replaced nature's elegance with 7 plants that we've designed. So what nature does is defend itself and manages to streamline that process naturally. We've clumsily sort of retrofitted. And I think it's important to consider this because the impact of synthetic biology is not just scientific and economic and industrial, but it's going to be ethical and personal and political and ecological. So this is the tree of life, or the current rendering of it, and with its 3 branches, we're down here somewhere with plants and animals. And this is a human-designed thing. It's a model that helps us make sense of the complexity of the living world. So while we were working on Growth Assembly, I was sitting in the studio and I was beginning to wonder, where would we put this stuff? How would we classify these things? Like, they might have genes from lots of these different kingdoms, or even completely synthetic, human-designed ones. And perhaps we'd need a new branch. So I added one, which is the synthetic kingdom. And the idea was that it was just, part of our new nature, nothing particularly significant about it, that it was just going to be an easy engineering solution to this problem of sort of classification, and just we'll carry on as we all are afterwards. What's been interesting about this for me is that it's been taken up in a way. This is the first textbook of synthetic biology. This is a journal that I have not read all of. And it was interesting putting this on the cover of these this is a journal which was a specialist on synthetic biology. And this image is, of course, it's a fiction. It's been a really useful fiction, and it's helped me have so many amazing conversations with scientists who willingly will suspend their disbelief to talk about something that they know isn't true. And so for this journal cover, I was on the phone with a Nobel Prize-winning scientist who was editing it. He was like, well, I think maybe you just need to move a couple of the labels. They're in the wrong place. It'll be more accurate if you do that. And suddenly we're having this conversation, which is just, it's so good. And others have said, well, I took it to a Tree of Life conference, and I was the pre-conference entertainment at 8 o'clock on a Saturday morning, and everyone started fighting about it. And they were, well, maybe it should be like little leaves like this, rather than one big branch, or maybe, well, it should definitely be smaller because synthetic biologists are never going to make that much stuff. My boss at Stanford is like, it's a big tabletop. He's a bit of a megalomaniac sometimes. He's like, maybe it's the whole, covers all the other kingdoms, or maybe it's more like spaghetti, like a network. I don't know which one of these is right. Probably none of them are. But what I do know is it's been a really useful tool to have discussion about whether designed organisms are different from what already exists, or maybe not that different at all. Some people have criticized me for giving synthetic biology a kingdom of its own and kind of validating it. But the question is, does this separate the things we design from nature more than we already do so? Or I think it puts our designs back into nature, and it removes that distinction between our things and ourselves. And if we can re-establish that connection between the things that we design and the ecological systems that they sit in, will we do better than we do today? So how do we take this kind of thinking into the lab? That was the next project. So after I graduated, I wondered what next. So this was the next iteration. Jim from Cambridge said, well, why don't you come up and join in iGEM? It was back in 2009. And at the time, iGEM, in its 5th iteration, 2004, it started like this. You've probably seen this photo lots of times. I'm actually in there, somewhere near the left at the top. That's 2010, and last year it was divided into regionals for the first time. So I'm going to show a video that we made retrospectively of the project for the Design Museum, and then I'll talk about it a bit more. EcoMI is an experimental collaboration between designers and scientists working in synthetic biology. In 2009, 7 Cambridge University undergraduates spent the summer learning the tools of synthetic biology, which is essentially a new approach to genetic engineering. Using standardized sequences of DNA in a format that's called biobricks, they learned to engineer bacteria. They designed their own biobricks using genes copied from existing organisms, inserted them into E. coli, and created bacteria that secrete colours visible to the naked eye. E-chromite went on to win the International Genetically Engineered Machine competition at MIT in 2009.
And joining us now is one of the winners of the iGEM competition. Welcome to Science Friday, Ms. Mullen.
Hi, thank you so much. I am part of the Cambridge 2009 iGEM team, and our project was called E-chromite. And what we were trying to do is to improve bacterial biosensors. They're bacteria that can tell you the concentration of a pollutant in water. And they can do this because inside them they have a detector. So we developed 2 different parts, the sensitivity tuner, and this actually tells the detector when to turn on and when to turn off. So you have control over what level of the pollutant you're detecting.
And how does the bacteria show that it's on or off?
We use something called a color generator, which means that our bacteria changed color when the detector got switched on.
Wow, so they light up in a different color.
They actually change color. It's visible to the naked eye.
So let's say if you put a swab of the bacteria in a polluted river, bacteria would just change color.
Yep, exactly. Though you'd probably want to put a sample of your water on a bacterial plate, maybe not the other way around.
Well, how would you envision something like this being used other ways in the future?
As designers, we worked with the team to explore E-chromite's potential as they were developing it in the lab. And together we imagined a timeline proposing ways that living colour could evolve over the next century. These scenarios, some of which are shown in this film, explore the different agendas that could shape E-chromite's use and in turn our everyday lives. One of the first real applications for this technology may arrive quite soon, a cheap disposable biosensor for testing groundwater contaminated by arsenic.
Bacteria could also be used to produce natural colourings and dyes. By 2015, there may be a profession of people who hunt for new pigments in the genes responsible, bringing them back for use in the food and textile industry. By 2039, you can go to the supermarket and buy this simple probiotic yogurt for cheap personalised disease monitoring. The yogurt drink contains E-chromite bacteria, which establish a colony in your gut. They monitor for chemical signals that indicate the presence of a wide range of diseases. If they detect a disease, they start generating the corresponding coloured pigment, producing an easily visible output to prompt you to seek your doctor.
2049 sees the rise of the Orange Liberation Front, a terrorist organisation from the Netherlands, who are angry because a biotech company in China has patented the gene for the colour orange.
In 2069, Google releases pollution mapping bacteria into the atmosphere that turn red in the presence of excess CO2. And as the saying goes, red sky in the morning, Google health warning.
Our collaboration meant that E-chromite was a technology that's designed from the start at both the genetic and the human scale, and with a long-term outlook. We found that design and science could have a meaningful exchange in the lab, which could prove useful when developing technologies in the future.
You know, I think it's a new term to most of the public, synthetic biology. You know, we've heard about all kinds of genetic engineering, stem cell research, but I think the phrase synthetic biology has not been used a lot lately. But I guess we're going to be hearing more of it.
Yes, I'm going to talk about one aspect of the project in more detail. It's the one everyone always wants to hear about. So we mocked up these poos in a briefcase, and we took them to iGEM with us. I was just saying to Fiona today, we did it because we couldn't afford the fee to present. It was about $800. So this was our guerrilla campaign to actually talk to the scientists there. What we wanted to do, this was the end of the project with the students, we wanted to challenge something that we saw in synthetic biology, which was this consistent representation of biology as machine parts, like this. The whole of synthetic biology, and it's changed a little, but at the time it was all about cogs and machines and computer chips and everything as a biological machine. But to us as designers, why is everyone being shy of the biology? So we saw the gut as a really ideal computing interface. It's a really interesting way to imagine a computer. And if this isn't what we want, then what do we want? So we took it to these scientists, and lots of them, there was some who wouldn't even look in the suitcase. It was amazing. But it's proved to be really provocative as an intervention. And something really interesting has come out of this. So this was 2009. And I've learnt that by imagining the future, even in a critical way, you may make it more likely. I don't know if what we made was too realistic, but I've had I often go to science conferences and see this image shown in presentations by scientists as something sort of ideal, as a kind of biological interface. And I've met several scientists who are trying to make this work. I've had phone calls from Yakult in Korea wanting me to sign an NDA to develop the technology. And I have to write, I'm sorry, it's a fictional technology. But I don't know if we got too close to reality with this mock-up, or if we ever actually slowly changed attitudes within the field. This idea of biology as cogs and machines is sort of, there's more of an acceptance of this biological interface now. But for me, what was most important in this was that we brought these kind of social implications into the engineering design process. The students were thinking about implications and applications at the same time. It was sort of inseparable in the lab during their design development. So this thinking sort of fed into this project, which was in 2011 with a lot of partners funded by the Wellcome Trust. We developed a 6-day pilot laboratory workshop for artists and designers and scientists to learn synthetic biology in the lab together. What was important here was that we designed protocols especially for this, that we commissioned, that had kind of narratives that fit into a broader cultural sort of theme throughout the 6 days of the workshop. And I wonder if this kind of model could be a better way for both science and arts and design to be taught, where the cultural context and the actual scientific learning are inseparable. So the next challenge is, how do we take these kinds of collaborative design science projects that question and innovate at the same time into a more real space? Is that possible? So I was thinking, like, where could I test these ideas out? And last year, I learnt that NASA was starting a synthetic biology initiative. Now, to go to Mars, you'll need 30,000 kilos of food and water for your 5 or 6 Mars astronauts. So NASA's hired lots of synthetic biologists to try and design novel organisms to be farmed by astronauts on the way to Mars. They were advertising for a food scientist, and I thought, oh, that's so good. Imagine designing food for space travel. But obviously I don't have a science background, but I thought there was something odd going on here, because mission success demands happy astronauts. 1,000 days of eating algae sounds to me like worse than death. Engineering solutions are one thing, but what about your emotional desires, like astronauts dreaming of steak? It's a really long way to Mars and back. So I listened into the space food conference that NASA organized. And on the first day, this woman at the beginning said, we really need to consider this mission success aspect and the psychological importance of, astronaut welfare. Don't just talk about algae. 2 days of algae, bacteria, and yeast discussions followed. So I was wondering, what is the ideal scenario, and would there be a way to actually collaborate with NASA and to do this? And it turned out not quite, but it's been an interesting process. So the project that emerged is called Seasons of the Void, and I worked with Sasha, who I did the plant project with, and Andrew Stellitano, who's a food designer. We were actually looking at the real research that's going on at NASA. So photosynthesis is the way things are farmed on Earth using sunlight for energy. But instead, in Seasons of the Void, our fruits gorge on electricity. So we imagined, as the ship flies away from the sun and Earth in the slingshot, electrosynthesis replacing photosynthesis. This is the growing tank where engineered yeast and bacteria grow into large fruits that are grown in the dark. Farming relies less on Earth seasons, but instead magnetic fields thrown out by solar storms that affect the ship's batteries as solar power is harvested to feed these fruits. The reason behind this what originally sort of intrigued me, is that advanced technologies that are designed for space often come back to Earth in a really mundane way. Things like cordless vacuums, baby formula, memory foam, these are all things in our lives that have been made glamorous and are really boring by space travel. And I wondered, if they solved this engineering functionality and the human sort of emotional need for steak and delicious food sort of equation, if we can balance that out, could these genetically modified fruits ever adopt the gloss of space travel and actually make genetic modified foods more palatable back on Earth. This is something that's been really intriguing me, is why has molecular gastronomy never stepped into this space of sort of extreme genetic modification for pleasure? So in a way, through the NASA's initiatives, GM may never have been further away or closer at the same time. So this is work in progress, and we're still not quite sure where it's going to go. I wrote to NASA this week to show it to them because they weren't able to collaborate with artists or designers because they may lose their funding. It was quite interesting. They can't be seen to be too innovative, which is not a problem for everyone, it turns out. So this synthetic aesthetics, which I'll talk about finally, a bigger project that was funded by the National Science Foundation and the EPSRC, which is the UK Science Funding Council, for about $500,000, were given to a project where we had 6 artists and designers and 6 synthetic biologists in residencies, in labs and studios around the world, just to ask questions. Heaven. Can you design nature? How do you design nature, and how would you design nature well? We got these artists and designers and scientists to crash their ways of working together and to try and find new questions. The idea was to bring critical discourse into the lab and the studio. So I'll share 3 projects with you briefly. The first, we paired an architect and a plant scientist together. You'd think they'd have little in common, but actually there was real overlap in the tools that they were using. Fernand uses 3D models to build models of his plants that he's looking at, and David uses genetic algorithms to design buildings. So they took xylem cells, which are the water transport system from artichokes, and they looked at how, as these cells grow, they solve these incredible spatial problems between themselves and their neighbors. So this just happens naturally, basically. So they wondered, could they actually collaborate with a computer scientist to find a way to extract this data for how these cells grow and effectively make a program that they could then apply to other structures to solve problems, like sort of making a design for a building structure. And what they were doing sort of fundamentally is challenging this core belief in synthetic biology that a biological computer means something built from DNA code. And instead of imposing digital logic on biology, they're saying, well, can we actually use natural logic as the thing we're trying to extract and take advantage of that? The whole cell, instead of just the DNA, becomes the computer. And they're presenting just a slightly different model for what a biological computer could be, but they're actually opening up new areas within the science and new ways of working as well within David's practice as an architect and in materials investigations. We paired 2 designers from IDEO with 2 cell biologists. So IDEO, I'm sure you all know, is a design firm famous for its design thinking. And 2 really interesting things emerged here. The first was the similarities between the scientific and the design process. As designers, we're used to this really messy process. Scientists are often amazed that we don't expect it to be a straight line of like design and deliver. But for scientists, they have this strange thing where that's what goes on in reality, but science has told us this straight story. Because it's the way it's reported in scientific papers. We did this, we did this, we did this. And everything else is, in a way, hidden. Science is creative and messy as well. And I think it's damaging that all this stuff is hidden. And it's a really interesting kind of idea to present to scientists, and they see truth in it. And the second thing was that for the designers, they were like, we're going to go into the lab, we're going to design all this biology, it's going to be amazing. And then they realized that's not going to happen anytime soon. Easy to engineer biology is not here yet. So they designed fictions they came out of workshops with the scientists, like this rendering of a cup they imagined that would grow its structure and it would have nutrients in it, and then you drink it and the nutrients would be released. But to me what was more interesting was this. Will and Adam couldn't resist refiguring the hello world into the IDEO corporate logo. And it's an extraordinary image. I think it really hints at what future relationships we could imagine as science is brought into a design firm and design thinking into the scientific laboratory. Could a firm like IDEO help define what good design means? Could we imagine sort of new kinds of research areas like MIT's Media Lab that bring design and biology together? I think this is probably the most provocative image from the whole project. Or maybe not. So The last one I'll show you is this project. We paired a biologist with a smell provocateur, Sissel Tollas. And they wanted to explore the cultural fear that they see between, this cultural fear of biology, this potential conflict between the antibacterial, pasteurized world that we live in today and this future that we're being promised is going to be powered by bacteria and we're all going to love bacteria so much. At the moment, we know this much. This is the human microbiome. All the bacteria that live inside us and on us, our own cells are outnumbered 10 to 1. And we're learning increasingly how much we need them to exist. And they're in cheese as well. So Limburger, I don't know if anyone's smelt or eaten Limburger, it smells like feet. Because there's the same bacteria that make your feet smell. I don't know why I'm pointing at my feet. The same bacteria that make your feet smell make Limburger smell. Bacteria can be made from the body. So when cheese is made, it's often made by hand. The curds are sort of stirred like this and bacteria come off them and add to the flavor. So the natural progression was to sample the human microbiome and make human cheese. I was lucky that I got to visit all of the synthetic aesthetics residencies. I learned a lot, but I learned about being in the right place at the wrong time because I got swabbed. Luckily, my armpit cheese was judged the best smelling, and Philosopher's Toe, my colleague, was the worst. But we're the quietest room that's ever seen this slide. It's amazing. Normally, everyone's kind of retching in the aisles. What's important here, though, is that this idea of context. So bacteria, where it is on our body or whether it's in our food, is really important. Why is it okay if it's on our bodies but not in our food? And what's been interesting for the scientists, they've responded the most kind of vigorously to this project. Drew, who I work with at Stanford, who's one of the founders of synthetic biology, for him, he was like, this changes everything. We are what we eat, but in a synthetic biology future, it may be that we eat what we are. We're closer to the bacteria than engineering diagrams portray. Human cheese makes our symbiosis with bacteria, makes it tangible and definitely smellable. So all of these different themes about designing nature and what it means to do this are addressed in the book that's going to be coming out. And I think this book is kind of unique because it's written by 20 different people, from architecture to music to smell to synthetic biology and everything in between. And we're really kind of doing something that I think hasn't been done before. Because it was funded by the scientific community. It means that these ideas feed straight back into the science, and that's been really unusual. And as well as informing the design and arts and policy discussions too, and I think it's this openness to ideas and questions and experimentation that's been the real achievement. So I've talked a bit about how I think using design in different ways can help us challenge what we thought or think as society are the problems that we face. And I've given some examples of how I can see that we can imagine bringing design and art earlier into the scientific process and the development of new technologies can help us open up new areas of thought. I think it can help us do more than just find better solutions to the problems that we face, but give us better problems to solve. We all know that the plastic bottle is a really bad idea and we still use it. It's the right answer. It sort of solves convenient drinking water, but it's the wrong problem. So what are the right problems to solve? And especially in synthetic biology, I don't know what they are yet. That's sort of the work ahead. This is about opening up the discussion. But this idea that if we ask better questions, we'll have better problems to solve, I think is really important if we are, as designers, hoping to make things better. Thank you.
How do you think your world as a designer sort of changed in that process?
I think for us we're just really curious about the science. My partner John especially is really into kind of getting his head around the science, and a lot of the scientists that we worked with, like this optogenetics and before that quantum physicists, is extremely difficult science. So we come, I think the conversation, I think we kind of we always have the design thing at the back of our mind, but we are so interested in learning more about their science. So there's a lot of, I think, just curiosity and sort of interest in their work, which helps start the conversation. I think that's quite important, because there have been lots of science art collaborations, and I have met scientists who are not, who go off like, but what happened to that project? I don't know, where was I involved? So I think it's how do you build a dialogue? I think that's for us.
I think that when I first went into the lab, it was, and said I was a designer, not an artist, it was about being seen as useful, and that was really, really helpful tool. So rather than being there because I was just really wanted to do something for myself, it was about this exchange of ideas. And a lot of it's been about working out what are the useful things to offer. And I think a lot of it is about mediation and finding and joining up lots of different groups of people. But it's also the willingness to ask questions that don't that seem really obvious, but actually offer a fresh perspective. So it's almost a sounding board for their own work. So sort of realizing things that I know sound really inane as a question, but sort of they hadn't thought of that because it's been about 10 years since they thought of something like that. So that's the refreshing aspect, I think.
So how is it for you as a designer? When you're going to leave the form, and when you go into the genetic code, for example, and you go more for that kind of functionality and leaving to design it?
I think that we're both still trying to find form. So the genetic code, in a way, I think what's interesting bringing design into this discussion is that if the scientists are sort of working at the genetic scale, we're still thinking about how that interfaces with people who might use it in the future. So everything I'm doing, I see that's where the useful expertise is. I'm thinking about the end user, even if they're a fictional person or maybe an animal, that it's about how we see this stuff and even exploring the aesthetics of it. So in a way, I'm less interested in the code itself.
Can you talk a little bit more about the fiction? Because you were saying before in your talk that, you know, using the fiction to start a conversation. Could that be seen as sort of misleading, or you've said that you find it as a good tool in which to start conversations?
I think that's the synthetic kingdom, because it is so clearly a fiction. I prefer to eChromai, which this catalogue was too close. So what I was saying, how the poo itself, because people can't even tell if it's meant to be a prototype, if it is real. I'm like, it's definitely not real. Or is it, it just looks too real, basically. That the line between it being a fiction or a prop or anything else is so blurred, but also it's made it so successful. But it sort of you then risk desensitizing people, whereas the synthetic kingdom, as a very abstract diagram, I think it's a very purist fiction. There's no way that unless someone had no idea about what the tree of life looked like. But I've mostly used it to go and talk to experts. So in a way, the fiction there's no harm, there's no risk of it sort of duping anyone, I think.
I think it's quite important, because we face that question all the time, and I don't know what the answer is. Like, some might call you a hoax artist. Like, at what point, where are the ethical boundaries when you start bringing fiction into reality? Like, John still gets emails about ARCing. I want to join ARCing. I've just been through a disaster. Or like when we did the Song of the Machine, we got emails from people saying, "So I am blind, and are you telling me I'm going to be able to see electricity? Are you going?" Like, there was this whole, and it's really hard. But I think it's a very important thing that we designers need to address now, because design fiction is becoming really popular. There are a lot of people who are making it popular, and I think it's a very valid question to ask. What are the tactics? How can we be tactical about finding the right language to talk about this? Because there's a danger that either you get pushed to the side that you're just doing this sort of thing as a sort of flippant exercise, or you just lied to me. And I think we face that a lot, and I think it's important. I don't know the answer, but I think we need to kind of start building a good vocabulary around it.
Daisy, have you had any kind of examples of that, where you've sort of done something more fictional? I mean, how far into fiction are you prepared to go with your projects, do you think?
Well, the new project, the space food thing, which only we finished a month ago, and I'm not fully, I think it needs another round of design. But the last image I showed, which the food designer Andrew made with the red, to this weird Martian landscape and the thing looking almost like ice cream, I find so intriguing because it's so weird. It's like I've never seen anything like it. And I don't know how, in a way I found that space really interesting, that there's an aesthetic there that's clearly not real, but it can help be a vehicle for the discussion. Because I think it was when the Yakult people emailed me, I just thought this has gone too far, that it sort of spread, it's got so lost from its original context as a project, which was about this intervention with iGEM and talking to the scientists themselves about the aesthetics of the technology. It's become so far, and it was in the MoMA Talk to Me show, and it sort of looked so odd there by all the digital machines that it's, unless you can actually control the whole story, I think it becomes really difficult to manage and work usefully with fictions.
But what happens when you go too far into fiction is that there are very little discussions happen outside of the world of design. And if we are trying to, like for instance, the poo, because people could connect to it, even though some thought it was real, you were probably able to have conversations with around people who would otherwise, if you'd done a pure fiction piece like The Thing, would look at it and go away, or some might think about it. But if it's that space, is that if you're really claiming to have discussions or creating pieces as discussion pieces, where are the discussions happening and who's? So I mean, those are the things we face every day as well in our practice. I think that's, I almost think it's great that Yakult is calling you, because you've managed to take a discussion piece into that space. I mean, yes, you have to create a language to be able to tell them that this is not real. I mean, James Auger's piece was in Time magazine as this tooth thing. I don't know. It's quite difficult.
But I felt that it lost its critical edge completely by the time, it's like, well, we can expect this technology. I expect in 20 years' time that I will be buying Yakult that makes my, that monitors me for diseases. Whereas the human cheese is real, and it's just as gross in a way.
Yeah.
But then what do you think about, a Minority Report as an influence of the future of gestural interfaces?
What do you mean, that we're nearly there? Or I think that's also the danger working in this space, that we don't know. I mean, not danger, but that's one of the, especially as designers start working more closely with science, that's kind of why I'm now asking, well, what do we, should we be being more strategic and tactical, saying, well, what is, with the plastic bottles, the plastic bags, those were people saying, well, this is a need that's been asked for, and we're going to solve that need. We need to make more economical carrier bags. So do we need to start looking in more depth at synthetic biology, as an example technology, saying, well, what is, where can we usefully apply the fiction so that they really provoke the science to move in the right direction? Then how do you judge that direction, and whose direction, and I think that's the really interesting next space for these kinds of things. So rather than just, when it becomes hard to tell whether they're real or not, do you want to actually be steering secretly?
I mean, it also is like Apple filed a patent for their for iPad or something, and I think Samsung managed to go and tell the core that no, actually this has been invented before in Stanley Kubrick's film Space Odyssey 2001, and based on that claim, Apple couldn't patent it. So Samsung succeeded in using design fiction, like a complete fiction, to say you've not invented this. This already existed in a film. So that's happened. So that's quite interesting, I think, just to be able to do that.
Any questions from the audience yet? Nico.
So very interesting talks. The one thing that I was thinking about all the time was there's an anecdote about during the Cold War when America and Russia tried to go to the moon. And they both needed a pen to write in space. And the Americans, it took them years to develop a pen that could write in space. the Russians took a pencil. But if it's not true, I'm sorry I didn't know that, but I think it's an anecdote that I wanted to bring up to make the point of how do you escape going down the wrong route and taking a long time to solve a problem that's kind of there, but you could solve it in a way more simpler way. Like, for example, with water bottles. Why not put up free water fountains everywhere so you wouldn't need the bottle anymore?
I agree, and that's one of the criticisms that's interesting around synthetic biology and iGEM, is that it's sort of technologies in search of a problem to solve. So rather, it's just trying to apply, it's like, oh, well, we've got synthetic biology, what can we solve with this? Rather than saying, is this the most appropriate technology? And I don't think you can isolate technology on its own. I think it fits in with the entire legal, political, patenting, intellectual property, and sort of economic system around it. So when you say, is genetically modified corn going to solve world hunger, it's not that simple. But we're not really allowed to challenge that. But the assumption is that's the only way to solve world hunger. Well, probably it will also include better farming and better food management and less waste, but that's not the way the economic system works. So while the water fountain is a good and a well-proven solution to avoiding water bottle use, it's not always the way. So in a way, the question is, can design be, as designers, we're expected to design the water bottles. So if we're not going to do that, what are we going to do? That's the kind of question I'd like to think about before I go to sleep.
No, I think that's okay.
So maybe first a question to you. Is the other space story versus Russia versus America the shower? Huge research into the space shower, and the Russians just had the wet cloth, using capillary force to not have flying water. Is that a true story? Because that's the other one I heard at some point. It's like, just take a cloth and wash yourself. I was really interested about the degree of speculativeness and how critical a project is that you were talking about earlier. And sometimes I have the feeling that, as you said yourself, you start losing interest when it becomes too real. People think it is real. That is in itself, to me, weird. Normally designers are quite happy when companies start picking up stuff that, oh, we could actually do this. Or, that's where it also can get really exciting, and also where you start to take responsibility for those speculations, right? If you stay in this satellite hovering, speculative bubble, foggy, sexiness out there, it never hits the ground and gets really messy. And of course, it's also very culturally biased. So obviously, you've showed your colored poo in the Puritan states of Great Britain and America, and that's like, ooh, armpit. And here's like, yeah. Our students smell like that. So in a way, isn't it more daring to also allow yourself sometimes to go down to that boring everyday implementation of something that was very crazy at some point?
I think it's a very good point. Yes, but I don't know. My personal interest in my own practice has been seeing how far up to the very origin of the kind of scientific, the stuff that's going on, can you go as a designer? Whereas, where does it just stop making sense? So I've been doing a collaboration with Sasha as well with complexity scientists, and I find that much harder. It doesn't seem like design what we're doing. We're trying to make a model of the whole world, and it's taken us 2 years, and mostly what we've done is air conditioned the gallery to the temperature of the weather forecast from the day before, and it's a very different way of working. And I don't think for me that's an uncomfortable practice, because it doesn't quite fit in the logic of everything else. I think there's lots of different ways of working. So within synthetic aesthetics, we had 6 different kinds of artists and designers. David, who's an architect, very practically minded. He wants to make biological materials. He started working with Autodesk. It is about making stuff and finding applications. And I think that's great. And Will as well, working at IDEO. Maybe it's less about applications, but it's about how can IDEO fit into the scientific research space. So much more applied. I think that's great. I mean, I just personally, maybe I prefer the hovering, it's safer, but also because I think I have the interest I have is looking at the future of design rather than making stuff. But I think it needs more designers to fit into those slots as well. I think it's a good observation.
I mean, we try and make that balance with our consultancy and lab, and I think it is quite satisfying. I mean, it's same like Daisy. We're very interested in the futures, in near futures area, and so we want to treat technology. We want to think of technology more as a social and a cultural construct rather than a problem-solving thing, activity. But if there are ways in which we could try and, for instance, a project where we're going to work with a group of autistic children to imagine new kinds of technology. This is fascinating because similar to the visually impaired people, you kind of have to make some things, kind of their constraints are very real and very, very tangible. And I am visually impaired and I want to go out and say hi to somebody and remember that person's name without feeling awkward and staying in my house for 25 days and not going out. So we do have to design that. But I think it's important that just like how the conversation of Google Glass has just moved into this practical thing of like, oh, now we've got fashion designers who are going to design the glasses and you can also have different colored bands, the conversation about actually our phobias, it's, this idea of technological defeatism, that, our phobias are, some people don't want this sort of thing, they're never discussed, they're not part of the conversation. And I think by doing this sort of work, you're raising the questions that are important before technologies become products and appear to have solved problems and become plastic bottles.
So I mean, I completely agree with that, and I sometimes describe when I'm trying to explain to someone what I do, I sometimes say it's applied bioethics, which is possibly the easy way out, but it's also, I think, within the scientific context that I work in, it's a useful descriptor because there's a reason to do it, but it's something I noticed in synthetic biology is that the synthetic biologists are saying, well, we're going to save the world, and this is going to be the technology that will do it, and everyone's going to be healthy, and they're not going to be hungry, and they're going to, it's all going to be amazing. And then the bioethicists or the social scientists are brought in, and they expected to offer the conflicting argument. Well, we should be worried about everyone dying, and everyone is going to get ill from this thing that escapes. And then there's nothing in the middle, and the argument immediately polarized just by the way that the discussion is set up. And well, probably everyone won't die. So the world will probably be saved. And so then that's the inevitable direction of progress, and it's sort of assumed that it will be okay, and so you can't challenge it anymore. And that's just a really interesting way that the discussion is set up. It's the polarity, like Drew calls it the half-pipe of doom, but it is ubiquitous. But it's what, to me, has been so interesting is that we're afraid of the same fictions that we were afraid of 60 years ago. So if you look at 1950s science fiction around biology, like The Day of the Triffids, or The Blob with Steve McQueen, like those are the same kind of themes of things going wrong, or like The Triffids, originally the book, not the film. It's Cold War technology that the Russians have been developing, and the Triffids escape, whereas in the film they actually come from outer space. But all those themes, the bio-error, the bioterror, those things were devised a long time ago, and we haven't found any new sort of aspects. So in a way, that's one application for this kind of thinking, is to actually uncover new questions that no one's thought of yet. So I think that's another aspect to it.
Does that guy's business card say "smell provocateur"?
And I don't think she has business cards. She wears her own smell. She describes herself as a scientist, artist, everything. But she's sponsored by International Flavors and Fragrances, and her medium, her studio it's all bottles of smell molecules. So she works through the medium of smell. But her main aim is to challenge the fact that we think that some smells are bad, where she says there's no such thing as a bad smell, only interesting ones.
I think that there's a very interesting, intriguing dichotomy in the work of designers who work with biology. On the one hand, you say there's the need to ask good questions, find out what is better, and you had one very good slide where you see that corn is made into sugar, and then you have the fermenters and make them into biofuels, and biofuels should be the kind of fuels that can be used in existing pipelines and existing engines and gas stations. So everything remains the same. Disruptive technology is not really disruptive in the socioeconomic way of things. Everything should remain the same, just technology may change, but not the players that use it and act upon it, are pointing that out, and probably also maybe in the future having some design objects that underline that distinction between on which level this disruption is taking place is one thing that I find very intriguing. On the other side, and this comes back to the occult and maybe also the NASA food that you showed in your last slide. It's a kind of, we had that in our last session here. It's kind of a diegetic prototyping, pre-product placement, like in the Space Odyssey. They came up with some ideas, and later on, some of them maybe they are going to be realized. So it's kind of, you're heading, you're like the avant-garde, the go west mentality, open up new spaces so people can start to think about what kind of products in the future might be there. In a way, you're kind of the forefront of the science and technology field in preparing the public mind for the things to come. In a way, so you're kind of a public relation institution, or can be used as a public relation institution. So from your experience, when you work with scientists and engineers, how do you think is your work being considered by these people? Are you considered as someone that comes in and helps them asking the questions that need to be asked, or are you more brought in as a way to deal with the public and sell the things in a colorful way so the gut feeling gets better with them?
Well, I resist that framing as much as I can. So this has been something that I've constantly had to push back on. And the space food project I wanted it to be a collaboration with NASA, and that didn't go to plan. So that project to me is sitting in a very strange and interesting space at the moment, and I think it needs more resolution because I don't know who it's for. It draws on existing research about electrosynthesis, and why not make it look nice or taste good? These are things that they're skirting around because in a way they need to make engineering solutions because they're NASA. So there's an interesting space there. So I don't know who that's for. And that's why I find that last image that Andrew made so unsettling, because it looks just like the ideobacterial plate. It's a normalizing and it's a PR tool sort of image, and that's why I find it uncomfortable. I think I describe my work less as public engagement, but as scientific engagement, which raises scientists, makes them uneasy. But I think it's important because I'm much more interested in challenging the science than getting people to accept it. But within science, as you know from your work, there is this attitude that the public are separate, they're kind of stupid. If they understood, they'd want it. Why can't they just understand why it's so good? And I think that scientists need to be more aware that they are part of society, and just they're so immune because they spend so much time looking at this stuff that when they step back, then these objects in a way are intended to make them step back and help them as useful tools to do that and look at the bigger picture.
I completely. I think that science is quite a glorified profession, and that everything that scientists do is amazing. It's for the cause of future of humankind, and there's very little questioning actually, and they are offering something tangible, they're offering solutions. And even to we are very small as designers, we offer very intangible things, and to actually go in and have a sustained dialogue with scientists is extremely difficult, because it's very difficult to explain to them why are we there, why are we interested, and actually why should they even bother having this dialogue over a period of time. So I think it may appear that there is the collaborations are few and far between. We might two kind of people showing project science is a very small sort of scale of actually how much science is going out there, and how much of that science and technology is being questioned in the way it's being developed, and for what is being developed, and what are the purposes. And I think we have a very important role to play in that kind of going in and asking those questions even to the scientists. And I wish there were more sort of sustained dialogues. I think there's a whole different genre of work on science communication, where communicators are trained to communicate science to the public. And I don't think, I also agree with Daisy in that we don't really do that kind of work.
But so from your perception, I'm pretty sure that both of you are not wanting to do science communication, but do you think that the people that you work with in science see you more as a science communicator or as someone to come in and ask difficult questions?
I think it depends on the scientist. So the collaborations that I've stuck with are the ones that are comfortable. And it hasn't been that difficult to find scientific collaborators who work in this way. I think that I would disagree with you on that, but maybe I've just been really lucky and identified the people, you know, and yeah, I think it could be that. But then I've done really strange projects, like I edited an issue of Current Opinion in Chemical Biology, which was a really interesting, terrifyingly crazy thing to do, to edit a science journal on aesthetics. So they wanted a special issue on aesthetics. I don't know anything about aesthetics. So in a way that was even more comfortable because it was purely for a scientific audience. And the hardest part was to try and get, the scientists were all keen because they wanted to be talking about these kinds of issues in their own journal. But it was getting artists and designers to contribute to that for something that was purely for a scientific audience that they found difficult. And was sort of, and also because it's all behind a paywall and it's, you know, it's sort of inaccessible research. So I think that, and I was asked to do that by a scientist, and that's an example where there's great interest, I think, to actually bring more of these ideas within and into science. And I think synthetic aesthetics was probably just a lucky chance that it happened, but I think it's been really influential in this sort of idea that this can happen within science rather than as artists and designers going in and then going out and facing the world with their work. What is confusing, I think, is that a lot of our work is shown in a public context to get awareness for it. So, you know, it goes into MoMA, it goes into museums and galleries, and it crosses over into art, so it's less clear what its function is and who it's for. And so it does start to look like public engagement, and it probably is as well. But that might not be the primary motivation for it.
So when you work with bacteria, do you think of them as living organisms? Because at iGEM you use them as machines.
I don't work with bacteria. That's
okay.
That's something I've chosen not to do. I've worked in laboratories a lot, and I've transformed things, but I've never designed a project using bacteria. I'm not sure if I ever will. I would like to, but I keep on finding myself not doing that because I don't know. I see them as alive, but not in a sort of, you know, they're not. I don't mind that they're engineered. I'm not in that space where it sort of seems something wrong with doing it. But there hasn't seemed to be a real reason for me to do it yet. But I do think this is something that's strange about synthetic biology, is that we can easily use living machines without really considering. Because as the things get bigger, the problems do, they change, like plants and how they interact with the ecosystem and the environment. It's a different set of considerations to bacteria and bats.
No, me neither. We're definitely much more interested in the wider political implications rather than working in the lab personally. So our projects, we're interested in placing them in the wider politics of our lived experiences. So I think that's been our focus so far anyway.
I think we should draw this to a close, but I'd like to thank you both very much indeed, Anab and Daisy. So thank you.